I'm Dr. Sarah Schlesinger, and I am probably the only person in the room more excited to be here than you are, and I'll tell you why. So I am an associate professor of clinical investigation and a senior attending physician here. As I like to tell people, I have only an MD and not an MD Ph.D., which is the degree that most people who do the work I do have. And the reason I have only the MD is because I'm really old. Back when I was training, a lot of people only got an MD. I think now most people would get both degrees, but I'm not going back. So it's really my pleasure to welcome you this morning to talking about science and introduce my friend and colleague, Dr. Jean-Laurent Casanova. I'm going to tell you more about Jean-Laurent momentarily. I'm going to try to be brief, which is not my strongest suit, but I'm going to tell you a little bit about the Rockefeller, about talking about science, and then a little bit about why I'm up here talking to you and my life, but only a little bit. So, the Rockefeller University is a remarkable, remarkable place. This is a photograph of it from, I guess, 115 years ago. We were founded in 1901, so you can note that there's almost nothing in front. The building that we're in didn't exist. When you come out, if you look, you'll be able to see those buildings as they existed then. They exist pretty much the same. Notice the 59th Street Bridge is in the background. That already existed, which is, I think, pretty cool. So the Rockefeller is first and foremost a biomedical research institute. We were founded as the Rockefeller Institute for Medical Research, and though we are formally a university, I think we are still first and foremost a research institute. We are a leader against fighting disease and the efforts to advance scientific knowledge in all its forms. The university is a leading graduate research university for doctoral candidates in biomedical sciences, chemistry, and physics. Students on this campus have already completed their undergraduate degrees, so you can't apply here for college. And they come to us to earn an MD and some MD PhDs. I mean, a PhD or an MD PhD, I apologize. There are approximately 175 graduate students. We're very tiny on campus each year in laboratories with more than 600 scientists who range from postdoctoral fellows to the 79 professors who head our university's labs. Talking about science is a very, very important part of our tradition of Rockefeller scientists giving back. This lecture first began in 1959, the year before I was born, and was presented as the Christmas lecture, and then as things got more ecumenical, became the holiday lecture on science for high school students. It was established by Alfred E. Mirsky, who was a biochemist here at the Rockefeller, and he was one of the pioneers of unlocking the secrets of the nucleus of the cell. Dr. Mirsky modeled the lecture on a popular lecture series of science talks given for young people established in London in the early 1800s by Michael Faraday. And they were given the day after Christmas, and they were called demonstrations at the time. And he was considered to be one of the greatest experimenters in the history of science. So in addition to everything else, we're all taking part in a very long-held tradition today. We're pleased to welcome over 50 schools from the 5 boroughs of New York and beyond to campus, and thank you for spending a precious day, I might note a snowy day, on a Saturday in the pursuit of scientific knowledge. May it only be the beginning of your careers and such. Now, in a few words, I'd like to tell you a little bit about how I got to where I am today. So I came to the Rockefeller campus through those gates on a snowy morning exactly 40 years ago, just as you did, to sit in this auditorium and hear a lecture that was amazing and changed my life. I recall it like yesterday, even though probably very few of the people in this room were born then. My excitement of sitting in the seats that you sit in today. And I cannot believe how much time has elapsed, nor would I have ever believed then that this institution would become my scientific home and that I would be standing in front of you to introduce Dr. Casanova. 40 years is a long time, and a lot has changed. In December of 1976, there was no internet, there were no personal computers, and the only portable phones that existed in James Bond movies. Students at the Rockefeller, I'm sorry, I have the wrong page here. So if you notice, This is the letter that I received when I was invited to come. I assume all of you registered on the internet. And there are a couple of things. Is this working? Yeah, can you see that? So note that the zip code is different. These were called the Christmas lectures. It was a guiding tour of the cell, which was wonderful. The lectures were after Christmas, and they were for 2 days. It was a lot of lecture. But I was told to call this number, no area code, between these hours, because that was the only way to reach Miss Lewis, or Mrs. Lewis. Phones didn't call people then, they called a place. And the person you wanted to reach had to be at that place. It was even before answering machines, at the moment at which you wished to reach them. This was the ticket I received. Did any of you receive tickets? No, I guess you just were on a list. And they were perforated. I guess they only collected the first section, and I kept it. This looks pretty much the same, except for the fact that now I'm doctor. And it was a wonderful lecture. So this is Dr. Christian de Duve, who shared the Nobel Prize. He was the person who I heard lecture. And he shared the Nobel Prize with Dr. George Palade for figuring out the microanatomy of the cell. He figured out where the organelles were. And that was a huge, huge discovery, and also it continues to impact science today. The lectures that he gave, which we were cytonauts, we toured the inside of the cell, were wonderful lectures and were compended in a book by Scientific American, which is still available online. I checked yesterday. And so I think that I would highly recommend those to you as well. So I came here and I was entranced. And that's Dr. de Duve later. Oh, sorry, I advanced too fast. And I was entranced. And I came home and I told my mother that I'd like to get a job here for the summer. I was expected to get a summer job. And before that, I'd been a camp counselor. Just by coincidence, at a New Year's party, so remember this is the end of December, my mother met somebody, a friend of a friend, who said her husband had the worst working hours of anybody she'd ever met. He was a doctor. He didn't even take care of patients. And it turned out he worked here at the Rockefeller. I had no idea what he did. I only knew his name. And his name was Ralph Steinman. I called him, frankly, with nerve that I would have only had when I was 16. Now, just for the record, don't call somebody here. We have a formal scientific outreach program, which is how young people now come to work here, which is headed by Dr. Jeannie Garbarino and her team. Jeannie, you want to stand up so everybody can see you, please? That's Jeannie. You can talk to her at lunch. You can find her. She has a whole online presence. So no anonymous phone calls, please. But Jeannie will be able to help you. But I called Ralph. We talked on the phone. And after he interviewed me sufficiently, he said I could work in the lab, but only he couldn't pay me. He was then and remained his entire life very frugal. So I started working in the lab in the summer of 1977 beside Ralph at the bench. He had just discovered a cell called the dendritic cell, which is the cell in the immune system that orchestrates immunity. How many of you have heard of them? Dendritic cells, anybody? Okay, well, if you haven't heard of them, they're in your textbooks, so I suggest you look them up. That's what they look like. That's from the original publication in 1973 and 1974. I'm wearing one on my lapel, which I'll be happy to tell you also about at lunch. When I came to work here, this was my ID. My signature's about the same. I look completely different. And notice they used my social security number, which we had to blur out, as my ID number. I didn't have the one from 77 and 78. Somehow I have this one. They made me get a new one every year. This is a picture of our lab. Can anybody find me? There we go. That's me. And that's Ralph from 1981. Like many discoveries, the discovery of dendritic cells was highly controversial. It took Ralph nearly 2 decades for it to be accepted, but accepted it was. And in 2011, he was recognized, or he and his work were recognized with the Nobel Prize in Physiology or Medicine for the discovery of dendritic cells and its role of adapted immunity. Now, I could go on and on and on about dendritic cells and on even longer about Ralph, but I won't. Those references, I think, have been provided to you in the further reading. But Today, we're not here to learn about dendritic cells. We're here to learn about infectious disease. And so that's a dendritic cell. And this is the Rockefeller on a sunnier day. So we're all in for a real treat here to hear Dr. Casanova. He is both a wonderful scientist and, in my view, a wonderful teacher. And I'm truly looking forward to his lecture. I'll be here all day. I'll be here at the break. When he's finished speaking, I'm going to come up again to discuss some housekeeping details about lunch and the activities at lunch. So please stay in your seats till you see me, and I tell you that we should all go to lunch. So thank you again all for coming, and without further ado, Jean-Laurent Casanova.
Thank you, Sarah. So thank you all. You don't look intimidated at all. I am terribly intimidated. I've given classes, lectures to graduate students, more rarely undergrads, but I've never taught a class or given a lecture to high school students. My track record is that I'm a father and a pediatrician, so I have interacted with teenagers, but I've actually never discussed scientific matters with them. So what we're going to discuss today is an idea supported by a wide range of facts, and this idea is that infectious diseases, which you know are infectious, that is caused by infectious agents, viruses, bacteria, fungi, or parasites, well, these infectious diseases, at least in some children, some adolescents, and some adults, are also genetic disorders. Not in the sense that the microbe is transmitted from the parents to the offspring, but in the sense that the determinism, the conditions that make some children or adults develop a full-blown, life-threatening infectious disease upon exposure to the infectious agent, well, these conditions, these determinism, are genetically determined. So that's the idea we're going to discuss today, and I'm going to take you very slowly to this idea and the data supporting it. We're going to have two sessions. During the first talk, we'll go from roughly the turn of the 19th century, that is around 1800, perhaps to 1950, maybe we'll go to 1990, I'm not too sure. And in the second talk this afternoon, we'll review the progress of this idea, its development, since 1950 or 1990, depending on how well we do during the first session. I would like to encourage you to ask questions during the talk, if it's permitted. Is it permitted? Where are the, yeah. Is that a yes or a no? I don't have my glasses. Absolutely. Absolutely, excellent. So yes, you see, you're encouraged to ask questions. It would help me actually if you ask questions. Even if you don't have a good question, talk to me. It's going to help me go through these 2 hours. All right. So I'm going to give you the version for grown-ups, but if you want the version for dummies about these ideas, I've published 2 papers in PNAS that review this idea, the genetic theory of infectious diseases. In the first paper, it's 1800 to 1990, and in the second paper, I review the data generated over the last 2 decades. If some of you have questions after the day, you can email me. I'll be happy to take your questions or advise you. All right, so we're going to go very slowly, and we're going to start with the beginning. I have no idea of your background. I didn't go to high school in this country. I went to high school in France. The programs are different, not only the language, but the programs are different. So I literally have no idea of what high school students learn in America. So you have to forgive me if I'm, pitching this talk with ideas that are obvious to you. If it's obvious, you just tell me, well, move on, fast forward, and I will. I can. Just let me know, okay? Good. So we're going to start with the beginning, of what science is. And science actually falls into two categories. There are the sciences of matter, or the sciences of inert matter, and the sciences of life, right? You're familiar with this dichotomy. Are you? You are. Very good. Now, there are 4 features of living organisms that make them fundamentally different from, say, this remote control, and that really account for the profound differences both in nature and in the discovery process between the sciences of matter and the sciences of life. First, living organisms sadly reach an end, and that's called death. That is, they live and they die, unlike, most inert objects. The second distinctive feature of life is that as a whole, that is the ensemble of living organisms, they evolve. And that also is very different from the inert matter. The universe, for example, can expand or contract, or transform, but it does not evolve. Whereas life does evolve. A third feature that is important to understand is that living organisms, such as yourselves, obey the laws of physics and chemistry. You're bound to your chair because of gravity, right? However, living organisms exhibit a variety of properties that their constitutive atoms and molecules do not exhibit. For example, you can attend this talk and understand it, but your electrons cannot do that. That is, living organisms can do things that their constitutive bricks cannot do. And this is often referred to, with the term emergence. There is emergence, that is, with each new layer of complexity in living organisms, there are functions that are generated by this new layer of complexity. Now, the fourth and very important and neglected idea that really makes a fundamental difference between living organisms and the inert matter is that all living organisms are unique. This is a very important idea. All the chairs on which you're sitting are identical, but all of you are different. Now, this is very important. It seems obvious. Maybe you're thinking, well, this is obvious. But, until the middle of the 19th century, nobody had understood that. And I can assure you that a number of eminent people today still don't understand that, including among biologists. This idea was expressed, verbalized simultaneously by 2 great biologists in the 19th century, Darwin and Bernard. For Darwin, the idea that all living organisms are different from one another formed the basis of his great insight that evolution occurs by gradual variation. And for Bernard, the idea that all living organisms are unique led to his notion that living organisms are self-sustained machines that should be studied as one organism and not as a population of organisms. And we're going to come back to this. Now, these 4 fundamental differences between living organisms, whether plants, animals, humans, and the inert matter, the reason I bring these differences is because these 4 elements will be important for you to understand the history of ideas in the field of infectious diseases and in the development of a genetic theory of infectious diseases. I also would like to stress that these 4 ideas are typically obscure to physicists and chemists. And when they try to understand biology or medicine, or when they join biology or medicine, the reason why in the vast majority of cases they fail making important contributions to the study of life sciences is because they don't really understand in depth the importance of these four notions, in particular the last two notions. Okay? Now, for those of you who are not good in math, I have good news, and that's the postscriptum here. It is that mathematics is not a science, it is a language, and it can be helpful to the conduct of science, including the conduct of life sciences, but it is not necessary. It is much less important, for example, than the command of English or French or Chinese or Spanish. Very great scientists have made spectacular discoveries, and they were innumerate. They couldn't count to 5. A good example is Charles Darwin, who cracked the most important problem without a single equation. So this is the life sciences versus the sciences of matter. Now, we're going to zoom gradually, progressively. Now I want to tell you a little bit more about the life sciences. And all these notions that are general notions, again, will be, I hope, important for you to understand what we're going to discuss throughout the day. Life sciences, in turn, also fall into 2 categories. I could have asked you, I don't know how many of you know that, but there are 2 types of life sciences. There's physiology and evolution. Physiology is the study of these self-sustained machines, the living organisms, again, whether plants, animals, or humans. It's the study of how living organisms work. And when they don't work, when they're sick, that's pathology. And pathology is, in essence, a derailment of physiology. That seems obvious to us now, but it was not obvious, as we'll discuss, by the turn of the 19th century. What is evolution? Evolution is the study of why living organisms work the way they do, and why they are so diverse, why all of you are so diverse, and why plants and animals, why are there millions of species on Earth. So physiology and evolution are the 2 branches of life science. We're going to focus mostly on physiology and pathology, but we'll allude to evolutionary questions from time to time during these 2 lectures. Now, before we delve into the problem of infectious diseases and their genetic basis, I have a final introductory slide to tell you about the importance of the history and philosophy of sciences. And that relates to how experiments, how a scientific program is being conducted. The joke that I tell my students, my grad students and postdocs all the time, and maybe they're tired of hearing me repeating this joke, is that there are millions of species, there are millions of organisms, millions of cells, and millions of genes, and that you can take randomly a gene, a cell, an organism, and a species, you can select them randomly, and you have a topic of research, you can study that combination. There are a sufficient number of tools that you can actually do science about anything. It's not going to make it an interesting scientific endeavor, but it is doable. So what I'm trying to tell you here is that science does not consist in doing things, in selecting randomly molecules, cells, organisms, and species. It consists essentially in selecting an enigma or a problem or a question, and preferably a good one, an important one. And throughout the talks I'm going to give you, I'll try to show you how our predecessors have focused their attention on important problems as opposed to unimportant problems. The second step in the scientific process consists in coming with an idea, a hypothesis, a solution, an answer, and here, it's better be the good one, because if you're going to spend 5, 10, 15, 20 years cracking a problem, you'd better start with a good idea. And 3, it's the approach. And this approach may be experimental, but not necessarily. The field of evolution in particular is not based on the experimental method. The field of physiology relies on experimental approaches, but the field of evolution is largely observational. And I alluded to that earlier when I discussed with you Charles Darwin. So, in short, for a scientist to do interesting and important science, it is very important that he or she is also a historian and a philosopher of science. Because the understanding of the history and philosophy of science, not only in his or her field, but more generally of at least life science, and preferably both life sciences and the sciences of matter, will help him or her select the good question, the question that matters, come up with the good hypothesis, and devise the appropriate approach. Now, after this long introduction, but that gives you, I think, hopefully a background to understand better the matters that we're going to discuss in more depth, I'm showing you in that slide the problem that I have been interested in for 20 years, and it is, in fact, a problem in which a number of great minds have been interested for a while. This problem is the problem of fever and death of children, infants, toddlers, children, and teenagers, also young adults. For 200,000 years, you know that human or species is probably around 200,000 years old. So for 200,000 years, worldwide, children would die of fever. And about half of the children would die of fever before the age of 15 years. So that life expectancy at birth was on average 20-25 years. That has been perfectly documented. We know that from the study of skeletons, from the Paleolithic and the Neolithic period, and more recently, from registries simply. So what you see here are mortality curves of our own species. And if you forget about the orange curve, which is the most recent, UK 2000, but it could be New York 2016 or 2017, what you see is all the curves are similar. And life expectancy on average was 20-25 years. Kids would die of fever. Typically, in a family of 5 or 6 children, 3 of them would die of fever at 2 years of age, 6 years of age, or 10 years of age. And that lasted until the end of the 19th century. That is, if we had been here 150 years ago, half of you would already have died. And it had been so for 200,000 years. So that, of course, was an important medical problem. And it is a problem that people have thought have cracked when microbes were shown to cause childhood fever and life-threatening infectious diseases. Evidence for that is shown with the orange curve. The orange curve, which is life expectancy nowadays, is around 80 years, maybe 83 or 84 for women and 78 or 79 for men. These results largely from the conquests that followed the discovery of the germ theory, which we're going to review in more depth. You wash hands, or you should. I always have a hand sanitizer with me. You were vaccinated. You hear, from some circles that vaccines are dangerous. This is very silly and very dangerous to say that, because vaccination results, has resulted in actually most of you being here today. So you can be very grateful to vaccines. And also antibiotic and more generally anti-infectious agents can kill microbes. That is, if an infectious disease has not been prevented by vaccination, and if it develops in any one of you, anti-infectious agents can treat you, and perhaps some of you have already been saved by antibiotics or other medications. And the fourth conquest that followed the germ theory is surgery, aseptic surgery. Until Lister, a surgeon who had read Pasteur's books, came up with the idea that surgery should be aseptic, most people would die during surgery or shortly thereafter of infectious complications. So infectious diseases are caused by infectious agents. And what we're going to discuss today, because that you know, what we're going to discuss today is that these infectious diseases, at least in some children and adults, are not only infectious disorders, but that they're also genetic disorders. But for you to better understand the development of this idea, of this genetic theory of infectious diseases, I'm going to take a step back and first review with you the development of the germ theory, that is, the establishment of the notion that infectious agents can kill. Do you have questions so far, or are you okay? Is it too simple, too complicated, too boring? It's okay? All right. Okay, your question here. So it's the use of lotions that are going to kill microbes. That is, if you have surgery and, you know, if you, for example, if you do not use gloves, if you do not use alcohol, the microbes will invade your body, and after surgery, you're going to develop infection. That's why the operating room is sterile, or as sterile as can be. All right, so now we're going to go to the late 18th century. Now, this is a painting by David. David was a great French painter. And this is the painting of Antoine Laurent de Lavoisier, who some of you may know was the founder of chemistry. He founded the entire field of chemistry. He showed that water consists of 2 atoms of hydrogen and 1 atom of oxygen. He actually coined the terms oxygen and hydrogen. You know, he realized the synthesis and the analysis of water. Anyway, he's the founder of chemistry. But what is less well known is that shortly before he was beheaded by the French during their revolution, the sort of things the French used to do at that time, very smart, he said, well, I would like to do a last experiment, please. And the jury told him, well, no, the French Republic does not need scientists. So he was beheaded, but shortly before that, he came up with a brilliant idea and a brilliant experiment. He showed that respiration, when you inhale and exhale, this is a combustion, and that it is a physical and chemical process. And he said, and he was the first to say that physiology, therefore living organisms, obey the laws of physics and chemistry. Had he not been beheaded for political reasons, he would probably have beheaded for having made that statement. So when he said that, he just had the time to train another eminent scientist, François Magendie. But before I tell you about Magendie, I want to stress that this painting, you can see it at the Metropolitan Museum. It was here at Rockefeller until 1976 or 77, but then the university transferred this painting to the Metropolitan. I really encourage you to go and see it. It is a beautiful painting. It's very mysterious in particular of why his wife is looking at the painter and he is looking at his wife. I mean, there are books have been written about this question. Anyway, it's an extraordinary painting. So he trained, he had this idea. He said, well, physicians are a little bit silly. They don't understand physics, and they don't understand minute discipline, chemistry. we have to educate physicians. Plus, they're pointless, they don't cure anybody. So what he decided was to take under his protection a few physicians and teach them chemistry and physics. And one of them was François Magendie. And François Magendie is not well known, even though he was the mentor of Claude Bernard, with whom you're perhaps more familiar. Claude Bernard being seen widely as the founder of experimental physiology and experimental medicine. And I want to read you here a quote from the In Memoriam that was written by Claude Bernard. This is Magendie here on the left, and this is another painting, I think by Fontaine-Latour, and this is Claude Bernard. Claude Bernard with some of his trainees a bit later in the 19th century. So Claude Bernard said, "My mentor Magendie exerted his influence on modern physiology by insisting throughout his life on the necessity of experiments. When he began this approach, he was alone in Europe. He was followed by and inspired many, including many foreigners. From his laboratory, young physiologists brought the seeds of new experimental physiology throughout Europe. The advent of experimental physiology took place in Magendie's laboratory. Magendie deserves the glory of having definitively planted the flag of experimental physiology." That's a nice tribute, isn't it? So what Magendie and Bernard actually did is that they proved that living organisms, animals and humans alike, were truly machines that were self-sustained and regulated. In particular, they showed that each organ had a particular function and that the whole organism was regulated with or thanks to the establishment of what they called the internal milieu, le milieu intérieur, which 50 years later, a great American physiologist, Walter Cannon, designated with the term homeostasis. So homeostasis, that is the equilibrium that is reached within living organisms, this is a notion that was established by Magendie and Bernard. Now, the reason why I take this detour is that when Bernard and Magendie established that living organisms obey the laws of physics and chemistry, and that everything in their organism ensures homeostasis, the idea that followed was that pathology was inevitably a derailment of physiology. The machine breaks. Therefore, when in these years a number of investigators proposed that childhood fever or severe fevers of adults were caused by environmental forces, they were not believed, because the emerging physiology and the emerging pathology really consisted in taking individual organisms as self-sustained machines independent from the environment. For centuries, the theories about human diseases, including fever in particular, were purely speculative, but people would think that disease were caused by mysterious environmental forces or by some kind of internal frailty. So with the development of physiology and pathology, the proponents of the environmental cause of fever were, in trouble. And I'm going to give you two examples of what were the consequences when some physicians proposed that that fever was caused by the environment. The first one is Ignaz Semmelweis. So he was a Hungarian obstetrician, and he worked in Vienna, who was then the capital of the Austro-Hungarian Empire. He was an obstetrician, and in these years, The problem was that 60 to 70 or 80% of women would die postpartum. They would give birth and they would die of fever. So he did a simple experiment. You know, there were 2 floors in his service. In one floor, he requested his interns and fellows to wash hands between each physical examination. They would see one patient, then they would wash hand, then the next woman, then they would wash hand, and so on. And on the other floor, they wouldn't wash hands. So on the floor where, you know, physicians would not wash hands, the mortality remained very high. But on the floor where physicians would wash hands, childbed fever disappeared. So that was a great discovery, of course, because he provided proof that childbed fever was caused by the environment, namely the doctor. And as a result of this discovery, you know, he was forced to resign. He was never hired by any hospital, and he ended his life miserably back in Hungary. Because the idea that fever could be caused by the environment was truly heretic. A second example is John Snow, another great mind who worked in London. And in these years in Europe, there were outbreaks of cholera. So he made a stunning observation. He noted that the outbreaks of cholera within the city of London, which, as you know, is widespread, were clustered around specific wells. In other words, the people would develop cholera if they would drink water from the same well. But there were regions, areas in London where there was no cholera. So that again indicated that this disease, this diarrhea, which killed, you know, high proportion of patients, was caused by the environment, was transmitted by water, was contagious. And nobody believed him. And he and Semmelweis are unsung heroes. Their discoveries really pointed to responsibility of the environment and therefore of microbes in the development of severe fevers of mankind. But for the reasons I've mentioned, in particular because of the importance of these nascent concepts of physiology and pathology, these ideas were not taken seriously. It took another man who was not only a great mind, but also truly a combatant, a fighter, Louis Pasteur, to establish the germ theory of disease. What you see here is Pasteur's family, his wife Mary and his 5 children. Now, 3 of his children, like so often in these years, 3 of these children died of fever, Jeanne, Cécile, and Camille. Incidentally, they had 5 children, like many families, because they gave birth at home. Giving birth in a service at the hospital that was almost guaranteed childbed fever, as we've discussed. So he had 5 children, he lost 3 of fever. Shortly after having lost his 3rd daughter of fever, he had a stroke from which he remained almost hemiplegic his entire life. He was in his late 40s. And he then was called by the French government to try to understand why silkworms throughout Europe, but in particular in the south of France, were decimated. So he went to the south of France, he built a hut, and he tackled the problem of the diseases of silkworms. And he discovered that the silkworms were killed by microbes. And that is the germ theory of disease. Shortly after having made this discovery, he identified the microbial causes of a variety of infections of animals and humans. It was a heroic period because, for the reasons I've mentioned earlier, nobody could think that these tiny microbes that were known, that had been first seen by Leeuwenhoek and that were known to Western societies, that these tiny microbes could derail big organisms such as humans, cows, horses, that really sounded heretic. And it took really him 20 years to convince the world, in particular physicians, that fever was caused by microorganisms. About 15, 20 years later, another heroic achievement convinced the world, perhaps more individually than any of the preceding discoveries of Pasteur, and this is the identification by Robert Koch, a German bacteriologist, that Mycobacterium tuberculosis, a microbe, a bacterium, is the causal agent of TB. TB stands for tuberculosis. This discovery had a huge impact because there were a variety of infectious diseases, but TB was the most dreadful. Everybody was afraid of TB, of having a patient with TB in his or her family in particular. Now, when Koch discovered the agent of TB, he attempted to clarify the field, the nascent field of microbiology, by defining strict criteria that would need to be fulfilled to assign the responsibility for any given human or animal disease to a particular microbe. And these criteria are referred to as the three postulates of Koch. A, the microbe occurs in every disease case. That is, if you want to say that Mycobacterium tuberculosis is the agent of TB, you need to prove that this bacterium is found in each and every patient with TB. Two, the microbe is not found otherwise. That is, it is not found in healthy individuals. And three, the third criterion is that the microbe has to be shown to transmit disease, not in humans. He would not inoculate humans, obviously, but in animal models. What's important for you to understand is that these three criteria that were very useful at that time to prove the germ theory and to identify rigorously causal microorganisms, that these three criteria indicated, falsely as we will see, that microbes are necessary and sufficient for the development of infectious diseases. Okay? Now, for you to better understand the impact of the discoveries made by Pasteur and then the identification of M. tuberculosis by Koch, I'm quoting here Theobald Smith, who was one of the first great physician scientists in this country. The building in which you will have lunch is called Smith. There's Smith and Flexner. So one of the two buildings, the Smith building, is named after Theobald Smith, who was one of the first physician scientists also working here at the Rockefeller. And in one of his very interesting books, Parasitism and Disease, that he published between the two world wars, he says the occurrence of tuberculosis in families led to the view that it was an inherited disease. The demonstration of a characteristic bacterium by Koch in 1882 disposed of this view. Here there are two ideas. One, the idea that it was a shock and a tour de force to prove that TB was caused by a tiny microbe. But there is also the idea that TB, and as we will see, other infectious diseases tended to segregate, to aggregate in particular families, which can be explained, in two ways. You can say, well, everybody has TB in that family because of contagion. You're contaminated by your relatives. That's one possible explanation. An alternative explanation is that relatives have TB because they share an intrinsic vulnerability and perhaps an intrinsic determinism to develop TB. That is, the disease, TB or any other disease, can be intrinsic or extrinsic. But in both cases, This can account for the familial aggregation of disease. Now, when I say that in 1882 everybody accepted the idea that fever, whether TB or other types of fever, was caused by infectious agents, it is not entirely true, because for decades there were resistant individuals, people who just did not believe the germ theory. And it will surprise you, but there are still people today who do not believe that microbes cause disease and who do not understand that vaccines are necessary. And I'm going to show you one example here. This is Max von Pettenkofer. So he was a southern German. He was working in Bavaria. And in these years, the northern part of the German-speaking world, there was Lutheran, and the southern part, typically Bavaria and Austria, that were Catholic regions, didn't get along too well, especially because, the unity of Germany was made under the rules of the Prussians. So people in Bavaria were not very happy about that. And one of them was Max von Pettenkofer, and he just hated the idea that Koch, who was a Prussian, would be right. So what he did one day, he did not believe in microbes. He took an entire flask of bacteria causing cholera, Vibrio cholerae. He took the entire bottle and he drank it, and he was fine. And therefore he published that Koch was wrong, because Koch had identified this Vibrio cholerae in Egypt after having identified the agent of TB. And, that was brave of him. Miraculously, he did very well. And he wrote that. He said, even if I had deceived myself and the experiment endangered my life, I would have looked death quietly in the eye, for mine would have been no foolish or cowardly suicide. I would have died in the service of science like a soldier in the field of honor. Man, if he will rise above the animals, must sacrifice both life and health for higher ideals. So he was a strong-minded individual, no doubt about that. And besides the anecdote, what is important for you to understand is that he was half right. Vibrio cholerae, the microbe of cholera, does cause cholera. But what he and others hinted at was that some individuals, the proportions of which being unknown, some individuals exposed to and even infected by agents that would kill others, some individuals would do well. He actually did. There's no reason to doubt that he didn't drink this entire flask and did not develop cholera. So the germ theory was solidly established. However, there were pockets of resistance. The real difficulty occurred a bit later, between 1905 and 1915, when two investigators, two great minds, probably my two role models, Clemens von Pirquet, who was a pediatrician in Vienna again, and Charles Nicolle, who was a bacteriologist in Tunis, discovered and proved scientifically, rationally, rigorously, that most individuals infected by most microbes are actually doing well and do not develop life-threatening infectious diseases. So Clemens von Pirquet is an interesting character. He was a pediatrician. He discovered allergy. He coined the term allergy. And one day he used the supernatant of Mycobacterium tuberculosis, the bacillus that causes TB, and he inoculated this supernatant that is this soup, of products that can be secreted by these bacteria. He inoculated this product under the skin of healthy volunteers who did not have TB, and he discovered, surprisingly, that they were all allergic to Mycobacterium tuberculosis, even though they did not have TB. That indicated or suggested strongly that not only individuals dying of TB, but also apparently healthy individuals in Vienna had also been infected by the agent of TB, although they did not develop clinical disease. What we've learned is that M. tuberculosis, a bit later, M. tuberculosis, like a number of other pathogens, viruses, bacteria, fungi, and parasites alike, can reside, I'll take your question in a second, can reside as dormant, non-replicating microorganisms in tissues. And the proof was established in 1927 when other investigators decided to test this idea, allergy to M. tuberculosis, fine, that's the tuberculin skin test that perhaps some of you have had. But these investigators in the late 20s decided to do autopsies on people dying of causes other than TB, and they discovered that from their lungs they could grow M. tuberculosis, people who never had any form of TB. That was evidence, compelling evidence, that the agent of TB could infect healthy individuals, not only people who develop TB. And if you recall, this observation directly contradicts the postulates that had been proposed by Koch. In other words, if Koch had done this experiment in 1882, he would not have considered M. tuberculosis as the agent of TB. Before discussing Charles Nicole, there's a question here. Question here, sorry.
I was wondering if the bacteria remained latent or did they become carriers like typhoid Mary?
So the bacteria, or some bacteria, some viruses, some fungi, and some parasites, they are the four big categories of infectious agents, can establish latency. That is, they can remain dormant, non-replicating within cells or within tissues. This is not a feature, it is not a universal feature of microorganisms, but it is shared by a large number of microorganisms. Now, the second one is Charles Nicole. So Charles Nicole, something tragic happened during his studies. So he was a medical student, and during his studies, he became deaf. He didn't know why, but he couldn't hear, which was a problem for a physician because in these years they had the stethoscope and that was it. There was nothing else. They could take a history, examine the patient, but they relied on the stethoscope. So he went to see Pasteur, and Pasteur, gave him a mission to go to Tunisia, which in these years was a French province. And he, told him, well, go to Tunisia, start a Pasteur Institute over there, and you can study diseases, infectious diseases that are prevalent in northern Africa. He discovered the transmission of typhus by lice, a great discovery that earned him the Nobel Prize. But what's interesting is that in his Nobel speech, he doesn't talk about that, or barely. His entire speech is about a discovery that he saw as his most important discovery, the discovery of unapparent infections. Now, an unapparent infection, according to him, is not quite a latent infection. In a latent infection, the microbe is dormant, does not replicate. He will, or there are conditions that may favor its reactivation at a later stage, but during latency, the microbe is dormant. What Charles Nicole discovered is that there were also healthy, asymptomatic animals and humans harboring rapidly replicating microorganisms in the bloodstream or in their tissues, which of course was even more paradoxical. And both the discovery of latent infection and the discovery of unapparent infections by these two pioneers clearly proved beyond any reasonable doubt that if microbes were necessary for the development of infectious diseases, they were not sufficient. And it gradually became apparent that most individuals, most plants, most animals are naturally resistant to most microorganisms, and that for any given microorganism, it is only a tiny proportion of infected individuals who develop life-threatening disease. The slide I'm showing you here is simply, again, to tell you a little bit more about Charles Nicolle. I took the two pictures on the left. I took them in Tunis. He was buried at the Pasteur Institute of Tunisia. And for those of you who have a good view, so Charles Nicolle was from Normandy. Normandy is a region in the northwest of France that is called Normandy because it was conquered by the Northmen, that is the Danes and the Swedes and the Norwegian, from where they then conquered Britain. Anyway, that's, they're Normand, okay, Northmen. Now, the tree over there is the apple tree. You have apple trees, miles and miles of apple tree. And in Tunisia, what's the tree? It's the olive tree. Now, on his grave, what you see here is this beautiful symbol of a branch of olive tree and a branch of apple tree on his grave at the Pasteur Institute. If you ever travel there, I encourage you to go and visit this place. It is very moving. Now, what you see here is a graph in which I have represented on the x-axis the proportion, the case fatality ratio, and on the y-axis, the number of infectious agents. This is simply a representation, some form of illustration and quantification of the idea that I've just discussed, namely that most infectious agents are harmless in most people. That is, that most of you have already been infected by microbes that have already killed others, or that could have killed others, and that you did okay, independently of a previous vaccination, of antibiotics, and so on and so forth. What you see here is that the bulk of infectious agents actually kill or can kill 1% or 1 in 1,000 or 1 in 10,000 infected individuals. You often hear that malaria is caused by a parasite called Plasmodium. Well, if you take any village in Africa, all children and adolescents have Plasmodium in their blood, in class, when they're playing, all of them. And it's only 1 in 1,000 children infected with Plasmodium falciparum who will develop life-threatening disease. And this applies to most, if not all, microbes you know. The microbes that kill more than 1 or 10% of infected individuals, they're very rare. There's HIV, there's Ebola, they're very rare. Most microorganisms at any given time kill or can kill only a very tiny proportion of infected individuals at a given time. Because as we will discuss later today, we evolve, so do microbes. That is, if Ebola and HIV ran, or avian flu or Zika ran out of control and killed 80% of the planet, well, in 100 or 200 years, the very same viruses would probably only kill a tiny proportion of the survivors by natural selection. In other words, the reason why, for example, malaria develops in only 1 in 1,000 infected children in Africa now is because these children have been selected for 200,000 years for resistance against Plasmodium falciparum. So the problem that is posed around 1910, 1920 is that microbes are necessary but not sufficient for the development of severe infectious diseases. The germ theory is correct, but the germ theory does not explain everything. Okay? Now, there are, from this observation, what time is it? Let me see. I probably have my watch somewhere. 11:40. When did we start? 10:30? Okay, so we have what? How many more minutes do we have for the first talk? You have to help me, okay? Remember, that's your mission. We have another, 10 minutes here or 20 minutes? How many more minutes? Until another half an hour? Good, good, good. I'm like Castro, I can speak for days. I could give a seminar on each of these slides. Well, I didn't harm anyone, unlike Castro. All right, so do you have questions before we discuss the solutions that were proposed to this fascinating problem? Do you have solutions? You have solutions, yeah, I hope you do, but do you have questions? I can sense that you need a break. No, there's a question over there. Yes, go ahead.
Does symptoms like TB also confuse the issue of inherited versus, being seen as coming down genetically as being taken by a microbe?
So, well, TB, like other conditions that had been identified clinically by their manifestations and also pathologically by analyzing, the histopathology, these conditions were thought by some to be intrinsic and by others to be extrinsic until the germ theory established that they were caused by microbes. But that lasted 30, 40 years. For 30, 40 years, the idea was that if you're infected, you're sick. If you're not infected, you're okay. And if you're okay, then you have not been infected. But this edifice collapses around 1910 with the discovery that, in fact, most people are infected by most microbes. Yeah, that's a good question. The question is, were there conditions that were shown to be caused by multiple microbes? I mean, not multiple times the same microbe, but two or more different microbes. So this is an interesting idea and notion that was not tested and documented in the 19th century. Evidence for this notion was established in the middle of the 20th century, for example, when it was shown that a high proportion of people dying during flu died of a superinfection by a bacterium. So this is not a discovery of the 19th century. It's a discovery of the mid-20th century. Yes. For example, when it was shown that a high proportion of people dying during flu died of a superinfection by a bacterium. So this is not a discovery of the 19th century. It's a discovery of the mid-20th century. Yes. Yes.
If you say in 1910 most people were infected with most microbes, that's no longer true, is it? Say in this room.
Well, it depends on how big you consider the ensemble of microbes, but we are infected, by the age of 6 or 7 years, we've been infected probably by 40 or 50 viruses, at least 40, 50 bacteria that can be harmful, a bunch of parasites and fungi. So yes, Pardon me?
So are they in us now or have we cleared them?
No, we don't clear them. So, for example, you carry a variety of microbes that can kill. You have Staphylococcus in your nostrils. You have Pneumococcus in your pharynx. You may have been infected by herpes simplex virus, CMV, EBV, and so you carry a variety of pathogens latent, in the latent phase or simply as commensals that with which you have established some form of peaceful agreement, but these very same microbes can kill others.
Okay. When do the viruses start coming in effect? Like, since we have them now, when do they start coming in effect and affecting us in the future?
When do the viruses come? Where do they come from? That's your question?
Yeah, when they come in effect.
So, how do they come? So, well, life, the universe is about 12 or 13 billion years old, and life appeared 3.5 or 3.6 billion years ago. And I think, I'm not a specialist of the evolution of viruses, but there are phages of bacteria, and that is of prokaryotes. So there's every reason to believe that viruses have occurred, have originated with the first forms of life, with the first prokaryotes billion years ago. And since viruses like prokaryotes and eukaryotes and archaea have evolved to generate, the millions of different species that we know.
You're saying that there's a sort of coexistence between humans and these kinds of bacteria?
Right. Well, that's right. There's a coexistence, as you say, exactly. There's not only coevolution, historically, but at any given time, there's coexistence, absolutely. That is, you carry commensals, that is, microorganisms that are going to feed from you or on you or within you, and some will be metabolically active and dividing. These are the unapparent infections, and others are dormant, latent. These are the latent infections. They do not replicate. Yes?
When bacteria become, that's loud, sorry. When bacteria become antibiotic resistant, does that mean the people who would be previously immune to it are no longer, or do they completely affect different parts of the bacteria?
So the question of the resistance to antibiotics or anti-infectious agents by microbes is a question I wanted to deal with a little bit later today, but this brings us to the question of your colleague, the evolution of microbes. So microbes evolve by natural selection, like us, like eukaryotes, like animals, but our own medical care can contribute to the selection of microbes. The more we use antibiotics or anti-infectious agents, the more pressure we exert on microorganisms, the more that will favor the emergence and spread of antibiotic-resistant microorganisms. Absolutely. Yes?
Sorry to drag on the questions, but so if you've mentioned the concept of a dormant infection that's like mutually beneficial, are there like environmental factors or bacteria or viruses that can trigger that infection after years of being there? Can it suddenly become harmful?
So your question is, what does trigger the reactivation? How do they wake up from latency, right? That's your question. Is that, yeah. So, well, one well-known example is when a patient develops an acquired immunodeficiency. He or she is dealing well with his commensals and latent or an apparent, replicating microorganisms, but then whether because he develops a malignancy or whether because he receives immunosuppressive drugs or is infected with HIV or another virus that is going to be immunosuppressive, then his defense will, weaken, and the microbe will take advantage of this immunodeficiency, reactivate from latency, and will cause disease. A good example is zoster. Most of you have probably been infected with the varicella zoster virus. During the primary infection, you develop varicella, chickenpox, then the virus remains latent. But if in the course of your life, and that can be 30 or 50 years later, you develop an immunodeficiency for any of the reasons I've mentioned, well, then the virus will reactivate from latency and cause zoster, which is some form of localized chickenpox, if you like, along a territory corresponding to the nerve in which the virus has established latency.
And acquired allergy work.
Pardon me?
Like, because if you repeatedly come in contact with, like, let's say latex, eventually you can be allergic to it. Is that the same idea? Because, like, latex accumulates in your body and somehow your body wants to fight it off.
Can you be allergic to an infectious agent? Is that your question?
No. How do we acquire an allergy to something we normally aren't allergic to?
How do you become allergic? Well, you know, this is a little bit off topic because we're discussing infectious diseases today, not allergy. Are you allergic? I'm allergic. I'm very interested in this question, but it's a little bit off topic. You know, in short, the molecular and cellular basis of allergy is not well known. You know, why you live with a cat and 2 years later, you know, you have asthma because of the cat and you have to, you know, give it to someone else. This is not well known, regrettably. Yes.
So, as I know that when doctors try to diagnose some infectious disease, they try to find either the microorganism in the body or either the antibodies in the body. So if you said that most of us have been infected by most of the microorganisms, then wouldn't we have a lot of positive, like fake positive diagnosis? Do you understand what I'm saying? So when you're trying, maybe I'm not correct in this. As I know that a lot of diagnosis is based on whether you can find a microorganism in the body or whether you can find an antibody in the body to see if you have that kind of infection.
That's right.
However, if you already get infected, you will have the antibody, right, surrounding your
That's true.
system.
Well,
so would we have like a fake positive diagnosis? That's my.
So, you know, the diagnosis of an infection can rely on the identification of the microbe or, as a surrogate, the detection of antibodies against that microbe, right? That prove, with hindsight, that you've been infected by this microbe, right? That's what you're saying. That's correct. And what is your question?
My question would be, if most of us has already been infected by most of the microorganisms, then if later day we develop disease, we try to diagnose it, the doctor will definitely find antibody from our body or find some leftover microorganism in our body. So even
Right.
if we were not get disease from that microorganism, we still got positive diagnosis. Do you
Right. Well, this is what, you know, the diagnosis of a condition requires evidence that you've been infected by the microbe. For example, if you do not have antibodies against M. tuberculosis, and if you don't have a tuberculin skin test that is positive, and if you don't find M. tuberculosis in the patient, you cannot claim that he has TB, or it's bold to make the claim. Conversely, a number of people are positive for antibodies or a skin test against M. tuberculosis, but do not have TB. The infectious agent is necessary but not sufficient for the development of disease. That's the idea.
For example, if a person have a symptom very similar to the disease caused by TB, all right, then the doctor will try to find TB from that person. Maybe the doctor will find it because that person was affected by TB. All right, but however, that disease probably is affected by other, by microorganisms, because TB sometimes got affected but doesn't cause the disease. Did you?
Right.
So
well,
is that possible?
This is why, yeah, the art of diagnosis can be difficult. It can be difficult to diagnose an infectious disease. For example, most of them are accompanied by fever. Many infectious diseases can affect the same organ. So in the absence of identification of the microbe or antibodies against the microbe, it can be very difficult. Yes. All right, one more question and then maybe we move on, otherwise we're going to spend a weekend here. Yes. IgG versus IgM. So the question here is, can we date the infection based on the nature of the antibodies? Yes, to some extent, the IgM, that's one type of antibodies, the IgM appear during infection and they disappear a few months after infection, whereas the IgG typically appear a little bit later and stay for a longer period. So that can help date the infection. I see that you have very practical questions. That's very good. All right. No more questions? Yes, one more. Maybe the last. Okay.
If I were to travel to a foreign country for a vaccine that is not native to the Americas, would it promote me contracting that infectious disease?
Right. Well, if you travel to a country that has infections not seen here, you should be vaccinated before you travel, if there is such a vaccine. If there is no vaccine, you should rely on hygiene. That's about it. Right? You wash hands, You be careful, with to whom you talk to. And more. All right. Other questions? Yes. Are viruses what? What? I don't. Are viruses alive? Do I have a joker? Are there questions I can pass? That. Well, you can sense that I like philosophy. Yeah, you're like me. This is a very difficult question. Lots of smart people have been thinking about it. So strictly speaking, no. But the problem is that now giant viruses have been discovered. There are viruses that are bigger than some bacteria. There are viruses that even encode transfer RNA, which was thought to be, the hallmark of living cells. And there are bacteria that cannot replicate because they have actually fewer transfer RNAs than these viruses. So they're obligate parasitic bacteria. So it's a very difficult question, frankly. That deserves, a conversation. Yes?
What do you have to say for those people who think vaccines are harmful? For instance, Jenny McCarthy doesn't vaccinate her kids because she believes it causes autism?
Well, she's wrong, and she's, No, I don't know who she is. I'm not American, as you may have guessed. Plus, English is not my native language, and so I don't know who she is. She's wrong and stupid too, obviously. But so you'll tell me who she is. Who is she? She's a Playboy model. Okay. Okay, so maybe I've seen her. Okay. All right. Anyway. Okay. I cannot hear you. You'll tell me more about her later. Okay. All right. Now, do we have more questions? Okay. Good. You're worse than the French teenagers. All right. So the problem that was posed around 1910 was that microbes were discovered to be necessary but not sufficient for the development of infectious diseases, okay? That's the problem. Now, this problem, if you think about it, there are 4 possible solutions to explain inter-individual variability in the course of infection. Someone is infected by the agent of cholera and is okay, and another one develops mild cholera, and the third one is going to die of cholera, even though they've all been infected by the same microbe. So that's inter-individual variability in the course of infection. So 4 ideas were proposed from these years onward. The first one is the radical version of the germ theory. That's the version that was proposed by microbiologists who are on stage and want to stay alone on stage. They think that the microbe is the cause and the only cause of infectious disease. So their way out is to say that if you die of cholera, it's either because you've been infected by more bacteria than your neighbor who's okay, or that the bacteria that have infected you, they were somehow different, that they had a mutation, that they were more virulent. And you can build such a theory. You know, you could say, for example, if someone comes here with flu and coughs again and again, at the end of the day, you can say that the 3 or 4 among you who developed flu, severe flu, you can say, well, perhaps they were the 3 or 4 who inhaled the biggest amount of viruses. You know, it's hard to disprove, it's hard to prove, it's hard to test. So there are still microbiologists who think that a life-threatening infection, when compared with an asymptomatic infection, is due to more pathogens or pathogens that are somewhat different from those infecting healthy individuals. Okay? So this theory makes sense. It's been documented in animal models. If you inject 1 bacterium or 1 million or 1 billion bacteria, clearly there's a different outcome, whatever the animal and whatever the bacterium considered. But in humans, I think it's fair to say that if there have been proofs that variability of infectious agents can account for the outbreak of a new type of disease, for example, the Beijing TB is caused by a new type of M. tuberculosis, but within a given auditorium or within a given city or a given region or a given country, the inter-individual variability cannot easily be explained by variations of the pathogen itself. There is still the possibility that some people are infected by more pathogens than others. This is true. So, and that may play a role. So that's one possibility. The second possibility, which again is an environmental hypothesis, is the idea that there are variations other than microbial variations that influence the infectious process. For example, you can say that if you're infected by rhinovirus, which is the virus that causes common cold, at -10 degrees or plus, I mean, I speak in Celsius, I'm sorry, so I don't know what it would give in Fahrenheit, you have to make the conversion. Let's say if you're infected by rhinovirus in the winter or in the summer, that's easier, then the outcome of the process would be different. That there are environmental changes that will impact the infectious process. So that also is plausible, and it has been documented. The problem, though, is that this does not account for inter-individual variability in a given environment, such as an auditorium or New York under the snow. It can account for the different outcome if you compare you today and today, some teenagers your age in Lomé, in Togo, okay? So, yeah, different environments. But it doesn't account for inter-individual variability in a given setting. So that brings us to the host theories. These two were the two environmental theories. Then you have two host theories. One is, I can refer to it as the genetic theory, and we're going to discuss it in more depth today. And the other, broadly speaking, is a somatic theory. The idea here is that in the years 1910, 1920, it was already known, as you pointed out, that people make antibodies against infectious agents, right? So a possibility that was proposed was that infectious agents act like some forms of mild or degenerate vaccines. That is, if half of you are vaccinated against flu, it is not a surprise that the cases of severe flu, if there were an outbreak, would preferably affect those who have not been vaccinated. So that was already known in 1910, 1920, because the discovery of vaccination is 1881. So the immunologists proposed that perhaps by cross-reaction, some infectious agents may trigger the secretion of antibodies that are going to protect against related microorganisms. For example, if you're infected by Staphylococcus, a particular type of Staphylococcus, maybe these antibodies are going to protect you from another type of Staphylococcus, and so on and so forth. So this is an immunological theory that proposes that inter-individual variability in the course of infection by any microbe reflects the past history of immunizations, artificial by vaccines or natural by infectious agents or allergens, to please your colleagues. So that is an immunological theory. It works, it is plausible in particular for infections that take place late in life. If you consider, let me try to explain that to you. If you consider a nursing home, people, they're in their 90s, okay, but they were brought to the nursing home from different, regions of the country. One lived in Alaska, another one lived in Florida, and the third had spent his entire life in Africa. And then at the age of 90, they all lived together in Missouri, for example. And there's an outbreak of something, flu. Well, they all have had 90 years of different experiences. They've been infected by different microbes for 90 years. And therefore, when all exposed to the same microbe in Missouri, their past history, as reflected in part by the titers of antibodies against a variety of different microorganisms, may help some and, on the contrary, be detrimental to others. So this immunological theory, the idea that our past or the memory of previous vaccinations and infections can account for inter-individual variability, it works best for the elderly population. If you now consider infections at a young age, a neonate, an infant, or a toddler, or a young child, it's much more difficult to take this hypothesis too seriously because they haven't diverged much. If you take 3 kids born in New York at 3 months of age, they've been more or less exposed to the same microorganisms, they've received the same vaccines. However, one day, One of them is going to end up in the pediatric intensive care unit with meningococcal sepsis, and the other one, who had been exposed to meningococcus, is going to be okay. So this immunological theory works best for the elderly population and not so well for infants and young children. Which brings us to the fourth theory, which is the genetic theory, the idea that a life-threatening infection, a severe infectious disease, is caused not only by infection, but also because there's a human genetic variation in the patient's genome that makes him or her vulnerable to that particular microorganism. And we will discuss this theory in more depth, in particular this afternoon. But what I want to discuss with you before is to tell you why this theory has been proposed, this genetic theory has been proposed by geneticists and not by microbiologists, the people who discovered and who still discover microorganisms, and not by immunologists, the people who discovered these famous antibodies that you mentioned earlier. The reason why microbiologists have not been studying this human genetic basis of infectious diseases is A, that they've been very busy with microbes, discovering new microbes, developing vaccines, developing antibiotics and anti-infectious agents. And B, as I alluded to earlier, that because they're the legatee of the greatest medical theory ever, they really see the microbe as being causal. And they often like to see the microbe as being exclusively causal of human diseases. So that's, in a nutshell, the history of microbiology. The immunologists, what operates is something a little bit different that I need a bit more time to explain to you. And that brings us to these famous antibodies. So in 1910, 1920, the immunologists know that individuals infected by a microbe make antibodies to that microbe. That's well known. They also show that if you transfer these antibodies to an animal that is healthy, and then soon to a human that is healthy, after proof of principle was provided in animals, that you can prevent the development of disease. In other words, these antibodies, these molecules that circulate in the blood and that can recognize an infectious agent like a lock and a key, well, these antibodies are protective. This is well known, and therefore, in 1910, 1920, the idea that antibodies are specific and that antibodies can protect a host, whether an animal or a human, this idea is proven. But what happens then at that particular moment is very interesting historically. In 1917, an eminent Austrian, Karl Landsteiner, decided to test whether animals can make antibodies against iPhones. And it worked. Very surprisingly, it worked. He synthesized compounds that do not exist in nature. He injected these chemical compounds, and he showed that animals, guinea pigs, or rats, or mice could make antibodies against artificial compounds, not only microbes that exist in nature. This discovery followed a set of experiments that had been done earlier by two other immunologists, Paul Ehrlich and Jules Bordet, who had shown, surprisingly, that antibodies can recognize plant toxins. Frankly, there was no reason to think that this would happen upon inoculation of plant toxins, but also foreign red cells, erythrocytes. Before these experiments were done, people thought that antibodies specific for infectious agents could be mounted by organisms because antibodies were predetermined to recognize infectious agents. So to note that antibodies could recognize plant toxins and then foreign erythrocytes, like a rabbit, for example, can make antibodies against a rat erythrocyte. That was very mind-boggling. But then Landsteiner took that to another level. He said, well, I'm going to create compounds that don't exist, inoculate them, and the animals made antibodies. This experiment that was done 100 years ago, exactly in 1917, this experiment blew all immunologists away, and frankly, they still haven't recovered, because since 1917, this is the only topic that has kept immunologists busy. And this is the antibody enigma. How is it possible that animals make antibodies against any structure? Okay? So in 1917, collectively, the immunologists said farewell to infectious agents and studied the antibody enigma. This is why, and here you have the two greatest immunologists, Pasteur, who started the field in 1881, and Landsteiner, who made this extraordinary experiment. So the immunologists left the problem of inter-individual variability in the course of infection. They've been busy with another equally fascinating enigma, the antibody enigma. So the microbiologists and the immunologists were too busy. And therefore, the immunological theory and the radical microbiological theory that I alluded to earlier were not really studied in depth by the microbiologists and the immunologists. It actually hasn't really been explicitly verbalized by these communities. Now, the third theory, if you recall, is the ecological theory, that is the environmental non-microbial theory. These proposes, if you may recall, that variations in the environment other than microbial variation in quality or in quantity can account for inter-individual variability in the course of infection. I gave you the example of the temperature and similar examples. Now, this theory was proposed by René Dubos, who was working here, like Landsteiner, by the way. And Dubos proposed this theory in a beautiful paper published in the Scientific American in 1955 with this beautiful title called Second Thoughts on the Germ Theory. It's a beautiful title, it's a provocative title, and the abstract reads, everyone harbors disease germs, yet not everyone is sick. This is ascribed to resistance, suggesting that germs are less important in disease than other factors affecting the condition of the host. Now, René Dubos was one of the founders of ecology. For example, he says, think globally, act locally. He wrote a number of very insightful books, essays. And in this paper, he elaborates an ecological theory of infectious diseases. He says that life-threatening infectious diseases occur because of variations in the environment that affect the host in the course of infection. Now, since this paper was published, I must say that this idea, this notion, has not been rigorously tested, and that even though there is anecdotal evidence that it may be the case, it has not blossomed into a solid theory. Although, yes, common sense suggests that these variations may be important. Which brings us to the fourth theory, which is the genetic theory. The idea that in the course of infection, the people who are very sick are the people who have a germline genetic variation, a mutation, if you like, that makes them vulnerable to that particular microbe. So this idea was proposed between 1920 and 1930 by a variety of geneticists, human geneticists, plant geneticists, animal geneticists, and both population geneticists and classical geneticists. We'll get later a little bit more into the differences between these terms and these communities. Here, the person I want to discuss with you is Archibald Garrod. Garrod was the founder of Mendelian genetics in humans, one of the founders of biochemical genetics, a great pediatrician. He had discovered the first Mendelian inborn errors, the inborn errors of metabolism. And he, after having discovered the first inborn errors of metabolism, he wrote a book in which he developed this notion of chemical individuality, which is one of the notions I discussed in the first or second slide of this talk. The idea that each individual is unique, each plant, each animal, and each human. He called that chemical individuality. And based on his discovery of inborn errors of metabolism and his notion of chemical individuality, he proposed that severe infectious diseases of childhood resulted not only from the invading microorganism, but also from mutations or genetic variations of the sick children or adolescents. So that is, in a nutshell, the genetic theory of infectious diseases.